Okay, now that we have our files organized in a collection here in test yeast, we can proceed with the first steps, which is the filtering. So the first step will be to trim the reeds either towards the right end or the left end. The reason is that the qualities of the sequencing drop towards the end of the alignment, towards the end of the sequencing, sorry. So let's see. So if this is a reed here, and we make a plot of the qualities along the length of the reed, then we will see that actually it's possible that it's something like that. Towards the 3' end mostly, the quality of the sequencing drops, and it might drop dramatically as well. Therefore, the idea is actually to remove this part and end with an alignment that's a bit shorter. But we will not cut just, let's say, 10 base pairs at the end of the reed. We will cut as many bases as needed if their quality is bad. And there are several tools to do that, to achieve that. We will use two of them, the Fastp and the Trimmomatic. And in order to check the quality of the reeds, we'll use again two tools. One is classic, the Fast QC, but now this is a bit outdated and it is replaced by another tool called Falco. Okay. Okay, so the report of Falco, the report of FastQC is similar, and now we can see the results after I have done some filtering that I will explain a bit later. So what we see here is on the x-axis the positions of the reeds. Our reed goes from position 1 to position 100. And on the y-axis we see the quality of the sequencing. So here we have done some filtering, and what we observe is that even at the end the read quality is quite high, is above 25, 26 actually. It's very high. So that's why we see green colors here. Everything has passed the quality filters. And you observe, however, that the quality of the sequencing drops towards the end of the reads. So for example, this box plot here and tell us that here at position 56, 57, the average quality is about 37. And we have the quartiles also here. So the first quantile is 34. The first quartile is 34.5, and the third quartile is 39. It's very high, and the minimum is 31, for example, which is also very high. Here, however, the quality is a bit lower. The median is about 35. It's not bad, but the minimum is about 26, which is not bad either, but not so high. Okay, so this plot shows how the reading quality changes according to the tile of the flow cell. So the flow cell, which is the device that sequencing is happening, is split in parts. So here we see 80 tiles. And this light blue here demonstrates the average quality. So for example, if we have a light blue line here, it shows that this tile reads all the bases more or less the same, with the same quality. If we see something purple, then maybe some tile is reading some specific base along the alignment, along the sequencing, along the read with lower quality. So for example, we see here that this tile here, which is, I don't know, something like 48 tile, reads the base at around 48 position or 49 position with a bit lower quality. Still, it's fine. So here we have the distribution of the quality scores for our reads, and we see that most of the reads have very high quality. There are a few that have lower quality, but they are just very few. Base per composition of the reads. So what we expect here is that the base per composition should not change along the read. Here it changes a little bit on the left hand side, but I don't see that something very dramatic. We can search Google if this is a bit problematic. Let's find some details. So here it's something that's happening usually in Illumina, and this might be related to barcodes or different other sequences linked on the left side of the read in order to start sequencing. But we see actually that the problem is minor here. The other thing that we observe is that we have 80 rich genomes. Perhaps somebody could check this if yeast is 80 rich. Since we see that 80 is about 30% and CG is about 19-20%. Okay, let's proceed. The GC content is okay. It's about 40%, similar to the theoretical distribution. We don't have many base pairs that are not that haven't been read along the sequence. The percentage here is very low, 0.25% here. For some reason we have slightly higher percentage around base 56 or 57. The distribution, most of the reads are 100 base pairs, which is the full length, but due to trimming we have done here, we have also some sequences that are a bit shorter. We don't have many sequences that are duplicated. Duplicated could indicate a PCR artifact. Here it's fine. So in general, and also we don't have we have poly A here, but we have cleaned for but still the percentage is small. We have cleaned for the adapters. Okay, so this is the quality of the reads after the filtering. We see also that our shortest read is 15 base pairs and the longest is 100. Perhaps we could put a threshold here and reject reads that are so short. We could maybe keep reads that are, I don't know, maybe 40-50 base pairs or longer. But since the yeast is a small genome, then even 15 base pairs could result in a precise match. Okay, let's go again to our files. So this was a Falco result. Let's see, and what is this here? Yeah, I want to just to see the FastQC that I ran, which was before the filtering, just to compare with the Falco results that are after the filtering, after trimming. Okay, let's see here. For example, you see here the distribution of qualities, and you see that towards the end, look here, there are several cases where the quality of the reads drops very, very low. Look, the qualities here. It's something like 6, 8, and so on. You remember the previous plot, our lowest qualities was something like 26, 27, some up to here. This happened because we had done some trimming and we had removed parts of the reads that were towards the right and the 3' end of the read. And the other plots are similar actually. This plot indicates that we should do some trimming towards the right end. This is exactly what Fastp does actually. Okay, I showed the results of Falco, which is a trimming with Fastp, and I have done some Trimmomatic also trimming, but I haven't talked about trimming yet. I just show the qualities. So in the next video, I will talk about trimming and see the options of Fastp and Trimmomatic.